Can somebody confirm that you can hear me?
Hear ya.
Thank you. All right, you should be seeing my screen now. Any questions before we get started? All right, I'm not hearing anything. So today is January 23rd, and we should be continuing chapter 3. Um, Let me inform you there is a lab today, lab 4 in stains. And our next quiz will be next Wednesday. That'll be quiz 2. It will cover chapter 3, the end of chapter 2, and labs 3 and 4. Any question about any of that? All right, I have that we were here. We discussed um, what a smear was, and that you make a thin film on a slide, and you make that before you do the staining. And then staining is just coloring the specimen or the background to increase the visibility by increasing the contrast between the specimen and its background. Any question about any of that? All right. A stain is a salt consisting of a positive and a negative ion. One of those ions will be colored, and that is what gives the dye its color. The colored portion is called the chromophore. Basic dyes have the chromophore in the cation, or the positive ion of the molecule. The acidic dye has the chromophore in the anion, or the negative part of the molecule. Any questions about any of that? Most of the dyes in microbiology are basic dyes, but we do have some acidic dyes. So dyes are salts. Bacteria have a slightly negative charge. Basic dyes are better for staining the cell because the positive cation will stick to the negatively charged cell. A crystal violet, methylene blue, malachite green, and safranin are all basic dyes. And these are very common stains used in microbiology. We'll talk about all of these in the lab today, or if you had the lab, we've already talked about them. And then crystal violet and safranin, we'll talk about them in this lecture as well as the lab. Acidic dyes typically stain the background, not the microbe. And that's because they have a negative charge to the acidic dye, and they will not stick to the cell. Some acidic dyes include eosine, acid fuchsin, and nigrosin. Staining the background of the slide instead of the cell is sometimes called negative staining. And this is because in the Polaroid negative, the light areas look dark and then the dark areas look light, and they call that a negative. And the stain kind of looked like the old Polaroid negative. Any question about any of that? So a negative stain stains the background of the slide, not the cell, because the cell is negatively charged and the dye is negatively charged. Any question about that? Here we're looking at two slides, each of them stained with a simple stain. This is a basic stain highlighting the cells, and this is a negative stain, which puts the color in the background. But as you notice, the cells are quite visible. And the reason why they're so visible is simply because a stain, all it does is increase the contrast between the cell and the background. And that includes negative stain as well. Any question about any of that? All right. In microbiology, we have three main types of stain: a simple stain, a differential stain, and special stains. A simple stain is an aqueous or alcohol solution of a single dye. The primary purpose is simply to increase the contrast between the background and the specimen. The stain is applied to a fixed smear for a certain length of time. The dye may then be washed off. It may be dried. It may be heat fixed. It's not in there. And then it may be examined. Oh, excuse me. You don't heat fix after you stain. You heat fix before you stain. Sorry. Um, the dye may be washed off. It's not always. The slide may be allowed to dry, and then it would be examined. You may have a washing step. Well, it's right there. Never mind. Not thinking well today. I did state that normally that's how a stain is done, but occasionally, like on a negative stain, you may leave the dye on the slide and then observe the specimen in the liquid dye in the on the slide. Any question about that? This slide has been negatively stained, but then the slide was dried. But like I said, you could look at the cell with the, you could do it with the basic stains too. You could look at the slide where the cells are sitting in the dye. Usually that's done with a negative stain, but sometimes it's done with a basic stain. Most of the time, the stain is put on, the slide is allowed to dry, and then you look at the slide. A simple stain may use a mordant. A mordant, all it does is it intensifies the stain. We'll talk about the mordant when we're talking about the Gram stain, which is not a simple stain. And iodine in the Gram stain is a mordant. It does not really dye anything in the cell. It just intensifies the stain of the first stain. The iodine, when crystal violet, it complexes with the crystal violet, making a larger molecule, increasing the affinity between the stain and the specimen, making the cell more visible, usually more intensely stained. Sometimes the mordant may simply coat the specimen to enhance it, thereby making it easier to see what you're viewing. Like in the flagella stain, it uses a mordant and it coats the flagella, and you apply the mordant, oops, several times. Let me get this in there a little better. And then there's many different coats on the flagella, and that'll allow the flagella to be viewed under the microscope. A simple stain, all the cells will stain with the same color. In a differential stain, usually the stain reacts differently with different kinds of microbes. And therefore you can distinguish between two types of microbes. Usually, two dyes are used in the differential stain, but that is not required. Most commonly used differential stains are the Gram stain and the acid-fast stain. Both of these differential stains do use two different dyes. Any question about any of that? The Gram stain was developed by Hans Christian Gram in 1884. You don't need to know when he did it, but you should know it was invented by Dr. Gram. And so we always capitalize the G when we say the Gram stain because it's named after Dr. Gram, and obviously his last name is capitalized. Any question about any of that? The Gram stain proved to be a very useful stain in bacteriology. He initially developed it to differentiate between the patient's cells and the bacteria cells causing the infection, which when he did it, the bacteria stained purple, and then the patient's own cells stained pink. So it was differential the way he was using it, but when microbiologists used it, they found it classified bacteria into two very large groups: the gram-positive cells, which stain purple, and the gram-negative cells, which stain pink. And nearly all bacteria can be classified into these two groups. The difference, we'll talk about this later in today's lesson, is the difference in the cell wall of the gram-positive cell from the cell wall of the gram-negative cell wall. To do the gram stain procedure, this is showing you the procedure in brief. You first make a bacterial smear on the glass slide. This is not doing anything with the stain. You are not doing the gram stain, but you need to have the bacterial smear before you can begin the gram stain. You then also want to air dry the slide and heat fix it so the cells stick firmly to the glass slide. If you do not air dry and heat fix the slide, then in the staining procedure, especially the washing steps, the cells will wash off the slide. And like I said, steps 1 and 2 is not actually part of the gram stain. You just need to make a bacterial smear before you begin the gram stain. So step 3 is the beginning of the gram stain. You take a bacterial smear and then you flood it, meaning cover the slide everywhere with crystal violet. And then you stain that for 60 seconds. You then wash off the crystal violet with cold water. You do that gently. We'll talk about that. And then you flood the slide with iodine for 60 seconds. Iodine is a mordant. It will intensify the crystal violet. You then wash the slide with cold water again. And then you flood the slide with acetone ethanol decolorizing agent for about 5 seconds. That will remove the crystal violet iodine complex from the gram-negative cells, but not from the gram-positive cells. And I say about 5 seconds because what you really should do is hold your slide up. Hopefully you can see this. Can you guys see this? I'm pretending it's a slide. Well, I'm going to assume you're seeing it because nobody's answering the question. And then you take your eyedropper of the decolorizing agent and you apply the decolorizing agent so that it runs down the slide. Once the crystal violet stops running on the slide, you stop applying the decolorizing agent. And that will happen in around 5 seconds. You then need to wash the slide immediately with water, and that's to remove all the acetone ethanol. And then you flood the slide with safranin, that's the counter stain, for 1 to 2 minutes. You then wash the slide with water again. You then blot the slide dry. Let me show you that. So this will be our slide. You put it in the bibulous paper. Usually you rip it out of the book, put it in the bibulous paper, fold it over actually. And then you just push the bibulus paper down on the slide. You do not move the slide against the paper. You do not move the paper against the slide because that will pull off the cells. You just blot it dry where you push the bibulus paper down on the slide. That will remove enough of the water. You can then apply oil and look at the specimen under the microscope. Any question about any of that? So when you're applying the gram stain, you first apply crystal violet, and all the cells will stain a bluish purple color. You then apply the iodine. Iodine is a mordant. It will complex with the crystal violet. It will intensify the stain. It will change the color slightly. Instead of a blue purple, it'll be more of a purple color. And you can't see either the intensity or the color difference in this slide, but just take my word for it. You then apply the decolorizing step, applying and usually an alcohol acetone decolorizer, although there are different decolorizers. You could just use alcohol alone, 95%, or you could use acid alcohol. Most of the decolorizers you apply for only about 5 seconds. It is true the 95% alcohol has to be applied for a longer period of time, something like around 20 seconds. Once again, when you're applying the decolorizer, what you should do is be watching the slide and then adding the decolorizer. And once the crystal violet stops running on the slide, you stop adding the decolorizer. Apply the safranin, it will make the gram-negative cells stain pink. Those are the decolorized ones. And the gram-positive cells will retain the crystal violet, so they will appear purple. Any questions about the step and the color? You do need to know this slide or the steps of the gram stain. Let's talk about the gram stain in greater detail. So once again, you start with a bacterial smear, which is air dried and heat fixed, and you flood it with crystal violet. This stain you apply for around 60 seconds. The exact time is not important. You could be a few seconds under. You could be a couple of minutes over, but you do not want the stain to dry on the slide. So around 60 seconds. You then wash gently with cold water, and that's to remove the excess crystal violet from the slide. The crystal violet will stick to the peptidoglycan in the bacterial cell wall. It's considered the primary stain since it colors all cells blue-purple. And it's the first stain you use as well. Any question about any of that? So the color of all the cells, both the gram-positive and the gram-negative cells, after applying the crystal violet, will be purple, and we can say blue-purple. You then apply iodine. Iodine is a mordant. It will chemically complex with the crystal violet, making an increase in the affinity of the crystal violet for the specimen because the crystal violet iodine complex is a larger molecule than the crystal violet molecule alone or the iodine molecule alone. You apply the iodine for around 60 seconds. Once again, the timing doesn't have to be exactly 60 seconds. It can be, I don't know, 5 or 10 seconds less, and it can be a few minutes longer. You don't want to have it so long, though, that the iodine dries on the slide. You then want to wash the slide gently with cold water to remove the excess iodine. Once again, the color of the gram-positive cells is purple. It's more purple color than the crystal violet alone, which was a bluish purple. And then the gram-negative cells are the same color, the purple color. Any question about any of that? The next step is the decolorizing step. The decolorizing agent we're going to talk about is acetone ethanol, but you could have a different decolorizing agent. The three most common, in fact, as far as I know, there's only three, is the acetone ethanol, and then there's acid ethanol, and there's also just 95% ethanol. The decolorizing agent washes the crystal violet iodine complex out of the gram-negative cells, but not out of the gram-positive cells. And that's as long as you do not overwash. It's also as long as you do not underwash. The decolorizing step is considered the differential step in the gram stain because after this step, the cells will appear different. As I stated, you apply the decolorizing agent for around 5 seconds, and the best way to do it is just apply it. Watch the crystal violet running down the slide. When the crystal violet stops watching, stop applying the decolorizing agent. You then need to wash immediately with cold water, gently wash immediately with cold water. All the other steps I didn't mention immediately because as long as the slide doesn't dry, you can wash with cold water whenever you want. Okay, obviously it has to be within, I don't know, about 5 to 10 minutes because otherwise the dye will dry on the slide. But in this step, you must wash with water immediately to remove the trace amounts of the decolorizing agent from the cells. Because if you do not, the trace amounts of the decolorizing agent will continue to work and continue to pull out the crystal violet iodine complex. And if given enough time, it will pull out the crystal violet iodine complex from the gram-positive cells, meaning you need to apply the decolorizing agent for the correct amount of time, which is around 5 seconds, so that only the crystal violet iodine complex is pulled out of the gram-negative cells. After applying the decolorizing agent, the gram-positive cells will stain a purple color, and the gram-negative cells will be colorless because the stain will be pulled out of them, the crystal violet iodine complex. Any question about any of that? You then apply safranin to the slide. Safranin is a pink dye. Safranin does stain all of the bacteria, both the gram-positive and the gram-negative. It's called the counter stain because it has a different color than the primary stain, which is the crystal violet. So what you do with the safranin is you apply it to the slide and leave it on the slide from 1 to 2 minutes. And then you wash gently with cold water to remove the safranin. And then you can look at the slide. You should wash it with cold water and then blot it on bibulus paper. And then you can look at the slide under the microscope, even if you're using oil. After the counter stain is applied, meaning after the safranin is applied, the gram-positive cells will stain purple because they're retaining the crystal violet iodine complex. And then the gram-negative cells will stain pink from the safranin. Any question about any of that? We already talked about that. So what is the basis of the gram staining cell? The gram-positive cells are staining purple by retaining the crystal violet. The gram-negative cells are staining pink from the safranin staining the cells which have lost the crystal violet. And why does this happen? What's the basis of the differential staining? The basis is related to the differences in the cell wall structure. The gram-positive bacteria have a thick peptidoglycan layer. We'll talk about peptidoglycan, well, I think in this lesson, actually. It's a disaccharide of many different chains, so really a polysaccharide built up on disaccharides and amino acids, making a layer in the cell walls. And this thick layer of peptidoglycan is very thick in the gram-positive cells. The gram-negative cells do have peptidoglycan, but it is a much thinner layer of peptidoglycan in their cell walls. The gram-negative cells also contain a lipopolysaccharide layer as part of their cell wall, and it's the outermost part. It's called the outer membrane of the cell wall, meaning it is part of the cell wall, but it's the outermost part, called the outer membrane of the cell wall. And there we're looking at a gram-positive cell, the purple being the peptidoglycan layers of the gram-positive cell, and it's a very thick layer compared to the thinner peptidoglycan layer of the gram-negative cells. And there we're looking at an artist's rendition of the cell wall. This layer there, that's the plasma membrane shown in yellow here or yellow there, and it's below the cell wall. And then we have the peptidoglycan layers. Only 3 layers are shown here. And remember, I mentioned that the gram-positive cell wall has about between 30 and hundreds of layers of peptidoglycan. Layers of peptidoglycan. And you can see some other molecules in the cell wall, including teichoic acid. Peptidoglycan is part of the, oh, never mind, that's the main molecule in the cell wall. And there can also be lipoteichoic acid. We'll talk about them in a little bit. Oh. The gram-negative cell wall has a much thinner layer of peptidoglycan. It also has a lipid bilayer outside the peptidoglycan, and then a lipid bilayer inside the peptidoglycan. The lipid bilayer inside the peptidoglycan is simply the cell membrane, called the plasma membrane here. And then we have our thin layer of peptidoglycan. Here, only one layer of peptidoglycan is shown. Usually on a gram-negative cell, there are between 30 and 20 layers of peptidoglycan, but only one layer is shown in this artist rendition. For that matter, on the gram-positive cell, you're only seeing 3 layers of peptidoglycan, and the layers of peptidoglycan on the gram-positive cell are much higher than 3. It's something like 30 to hundreds of layers. Oh, there we go. You can see that the gram-negative cell wall is different than the gram-positive cell wall, and there are more differences just than the thickness of the peptidoglycan layer. The gram-negative cell has some different molecules in the cell wall, as well as it has a membrane outside the cell wall. And this membrane is called the outer membrane of the cell wall. The main molecule in the outer membrane is the same main molecule is in the cell membrane, and that's phospholipid. Let me see if I can blow this up. That's better. So right there and right there is the phospholipid, and it's mostly a lipid bilayer, phospholipid and largely phospholipid in the second layer as well. It's not all phospholipid, especially in the outer membrane, but it's the main molecule in both the cell membrane and in the lower layer of the outer membrane, as well as in the upper layer of the outer membrane. So phospholipid is the main molecule in all of these membranes. You can notice that the outer membrane does have some other lipids besides phospholipid. The inner outer membrane does not have this lipid. It only has the phospholipids and other molecules in the outer membrane, such as carbohydrates and proteins. Here's a protein going through the outer membrane, and this protein is actually inside the cell wall and attaches to the cell membrane. So there are other molecules in the outer membrane. The one I want to mention the most is lipid A. Lipid A is only in the outer layer of the outer membrane of the cell wall. And I should say a gram-negative cell wall as well. The lipid A does have an O polysaccharide attached to it. Together, the lipid A and the O polysaccharide are called lipopolysaccharides. Lipid A is important because it is an endotoxin. This means that anyone who has a gram-negative bacterial infection, as long as the bacteria have a cell wall, they will contain lipid A in their cell wall. And toxins, if you do not know, are something that are bad for us because toxins are toxic to us. And so anyone who has a gram-negative bacterial infection with a cell wall will contain lipid A in their cell wall. And the patient's going to have to deal with being exposed to that endotoxin. Clinicians will want to kill off the bacteria, which will release the lipid A. But the worst thing to do is to let the bacteria grow until there's even more of them, and then the patient will have a higher exposure to lipid A. Any question about any of that? All right, where do I see that? I don't see it. There are some other molecules in the cell wall of the gram-negative cell, like the porin protein, the lipoprotein, and some other proteins, but oh, there it is there. I'll talk about that. So let me put that there. So the point is there are different molecules in the gram-negative cell wall. You never find the outer membrane of the cell wall in a gram-positive cell. You never find lipid A in a gram-positive cell. You don't find porin molecules in a gram-positive cell. So you have the porin protein, which forms the porin channel. The porin proteins are important for a gram-negative cell because this membrane is a bilayer, and bilayers do not readily let molecules either into the cell or out of the cell unless there's a protein to move it from one side of the membrane to the other. The porin protein acts as a non-specific or kind of a specific channel protein, allowing many different molecules to enter the outer membrane of the cell wall. So something like an amino acid or glucose or fructose, that can get through the porin channel protein. And then it can get through the peptidoglycan because the peptidoglycan is water soluble. So any water soluble molecule can go right through the peptidoglycan layer. And then when the molecule reaches the cell membrane, if it cannot get across the lipid bilayer of the cell membrane will make a protein, which will be a transport protein, either usually facilitated diffusion or active transport, to bring that molecule inside the cell. So something like arginine, an amino acid, the cell is going to want it, and it can non-specifically come through the porin protein and then come to the cell membrane because it goes right through the peptidoglycan layer. And to come inside the cell membrane has to make a transport protein that essentially can move one molecule, in this case, what did I mention? Glucose? Or did I mention an amino acid? I think I mentioned an amino acid like lysine. So the amino acid like lysine will move through the porin protein, move through the peptidoglycan, come up to the cell membrane. And then for it to move inside the cell, it has to move through a transport protein. And the cell needs to make those transport proteins and put them in the cell membrane to get them to work. Any question about any of that? All right, the basis of the Gram stain is the crystal violet iodine complex forms in all cells. And the crystal violet iodine complex is larger than crystal violet or iodine alone. And during the alcohol acetone wash step or the decolorizing step, the gram-negative cells have the crystal violet iodine washed out because of their thinner peptidoglycan layer. If you're wondering, the lipopolysaccharide layer, meaning the outer membrane of the cell wall, will be disrupted by the alcohol wash. And so therefore, these cells are colorless until you apply the safranin. The gram-positive cells have the crystal violet iodine complex in a thicker cell wall, so it is not, meaning the crystal violet iodine complex, is not washed out of the thick peptidoglycan layer. So the cells stain purple. So gram-positive are purple, gram-negative are pink. You were looking at a gram stain, a gram-positive coccus, a gram-positive rod, a gram-negative spiral, or spirillum, and a gram-negative rod. Any question about any of that? There are no gram-negative cocci in this specimen. Oops. Any question about anything? Shrink that down a little. All right, the gram stain doesn't always work perfectly. Like right here, these cells are real light purple, pinkish in color, and these are really dark, and all the cells next to them are dark. In fact, all of the cells in this region are darker than outside that region. Maybe right there too. So what do you do there? What you do when you get something like this, oops, to move the wrong way, is you do not stain the bad regions of the slide, meaning you do not look at the bad regions of the slide. That right there, as well as there and there, is actually a crystal of the crystal violet stain. And when you use the bottle, particularly on the bottom, there will be crystals of crystal violet. And if you put those on your slide, it will foul up that region because that's a crystal violet crystal. And then it'll make the cells around it darker because the crystal violet comes out of the crystal and then starts staining the cells nearest to it. Why these cells are much darker than those cells there. For the Gram stain, the most consistent results are obtained when performed on young growing bacteria. If your cells are greater than 2 days old, you should not apply the Gram stain because an error may result if the worms have been if the slide has old cells on it, the crystal violet iodine complex will get in the cells, but on the gram-positive cells, if the cells are dead or decaying, then the crystal violet iodine complex can get out of the cells because the cell wall is not retaining the crystal violet iodine complex. So it's acting like the cell is thinner, has a thinner peptidoglycan layer than it should. And then some bacteria cells stain poorly, if at all, with the Gram stain. It's mostly the mycobacterium that have a lot of mycolic acid in the cell wall. Mycolic acid does not like many of the stains to come in and then stain the cell. And the reason being, I guess I didn't talk about that. Let me talk about that. The reason being is in the gram-positive cells, anything that's water soluble can get right through the peptidoglycan layer. In the gram-negative cells, shown here, that outer membrane can keep molecules from entering the cell. And if the cell wants the molecule, that molecule has to come through the porin protein of the outer membrane and then a transport protein of the cell membrane. Where were we? The results of the Gram stain are not always as clear cut as theories suggest. If you were to do the Gram stain, you would learn that it's a little tricky procedure and it does take some practice to learn it. Structure of the gram-positive and gram-negative cell walls not only affects the stain, meaning whether or not the crystal violet stays in the cells or not, it also is related to how well the cells respond to certain antibiotics. Gram-positive bacteria tend to be easily killed by penicillin G. The reason, remind me that I'm at 52. The reason being is that penicillin G can get right through the cell wall and then do its damage to the peptidoglycan so that the penicillin G can easily kill off the gram-positive cells. But on the gram-negative cell, let me blow this up, the penicillin G cannot get through the outer membrane of the cell wall. If the penicillin G is on that side of the outer membrane, then the penicillin G cannot do damage to the peptidoglycan layer of the cell wall. So if that doesn't happen, then penicillin G will be ineffective on treating a gram-positive bacterial infection. Any question about any of that? So the difference in the cell wall can have more than just how the cell will stain under the gram stain. It can also affect certain antibiotics and other agents that could kill cells. Oops, let me get out of there. Gram-positive cells, besides penicillin G and penicillin V, can also be killed by cephalosporin and many detergents. Gram-negative bacteria are resistant to uh, penicillin G, cephalosporin, and at least some detergents because these molecules cannot get through the porin protein in the outer membrane of the cell wall. And if these agents cannot get through the outer membrane of the cell wall, they cannot go in and damage the cell. And the reason is they simply cannot penetrate the lipid polysaccharide layer, meaning the outer membrane of the cell wall. That doesn't mean that gram-negative cells cannot be killed by antibiotics. You can use a antibiotic that's chemically modified or made in a lab to enter the gram-negative cell. And ampicillin is an antibiotic which kills both gram-positive and gram-negative cells. And the reason being that it has modifications made to the molecule. So now the molecule can get through the outer membrane of the cell wall to do its damage. And then there's other antibiotics like streptomycin, which are lipid soluble, and they'll get right into the outer membrane of the cell wall because they're lipid soluble molecules. Any question about any of that? All right. I'm going to talk a little bit about the special stains because you may need them for your unknown project in the lab, but I'm not going to quiz you on any of this information, so you will not be examined on special stains other than what I've already talked to you about. A capsule stain is a special stain used to look for the cell's capsules. Cell capsule is something secreted by the cell to make many layers of this secretion around the cell. And if you have many layers that are more than water, then we call it a capsule. And you can view the capsule under a light microscope. There are other glycocalyxes besides the capsule. One of them is the slime layer. The slime layer cannot be seen under the light microscope. To see the slime layer, you have to have an electron microscope. The reason being the slime layer is mostly water, and so it just does not show up well with the Gram stain. Anyways, here's a negative stain staining the background of the slide. That's just the glass. And it's staining the cells as well. And you'll note that there's a region around each cell that is not staining, and that's the capsule. Right there, well, let's go there. Right there, we probably have a capsule, which has come off of a cell, because I don't see a cell in there. The same with that one and that one. And then there's a more common negative stain. This is staining with India ink. The India ink mostly stains the background of the cell because it's a negative, not a negative, a negative staining chromophore, meaning it's an acidic stain. Anyways, it's staining the background of the cell. It's very lightly staining the cell gray. And then around the cell, we have a region that is not stained, and it's appearing white or clear. And that's because that's where the capsule is. It does not take up the stain, and then you can view the capsule. Any question about any of that? All right, the capsule cannot be seen with most stains because the procedure will wash the capsule off the cell. You have to use the capsule stain in order to see the capsule. All right, any questions about this lesson? If not, I'm done. I think I'm finished, but let me check here. Yep, I'm finished with chapter 3. So that's it for the lesson. If there's no questions, I will say goodbye and I will see you next time. Some of you I'll see in the lab tonight, the regular time.
Thank you.
All right.
Thank you. Have a good night.
You too.
Have a good night.